Okay, good afternoon, everybody. Welcome to the CDCR seminar. And I'm really delighted today to welcome my friend and colleague, our own Professor Anthony Rullo. Dr. Rullo, do you prefer to be called a chemical immunologist or an immunological chemist?
I never thought of the latter.
Okay. So we'll go with the first.
I like the research.
I like them both. I introduce you variably as one or the other, depending on how I feel. Yeah. So in any case, I want to tell you a little bit about Dr. Rullo, who is an assistant professor of medicine and chemical biology at McMaster. He's one of the world's leading pioneers in small molecule proximity-inducing therapeutic strategies with applications to both immuno-oncology and antiviral immunotherapeutics. So in 2018, we were lucky to have Dr. Rullo found his lab here at McMaster University, and it's a translational chemical biology lab, which is very brilliantly integrated into the Department of Medicine. And I think we all benefit from the fact that Dr. Rullo is not isolated in the chemistry building where we could never find him. It's lovely to have chemists embedded in the Faculty of Health Sciences to converse directly with people to generate solutions to healthcare problems. So it's really great to have him right here, and of course appointed as well to the CDCR as one of our faculty, and also to the McMaster Immunology Research Center. So Dr. Rullo's scientific training encompasses a lot of aspects of organic synthesis, carbohydrate chemistry, physical biochemistry, and bridges to immunology. So I think he's a great example for all of you trainees in terms of looking at how someone can build their own expertise in multidisciplinary science and how bridging different fields actually probably better leads to new discoveries. Dr. Rullo got his PhD in organic and biochemistry from the University of Toronto under the supervision of Professor Mark Nitz. And there he developed fluorescent chemical probes of complex glycoaminoglycan binding interactions, in addition to new bioorthogonal affinity labeling strategies. And this was followed by a postdoc under the mentorship of Dr. David Spiegel at Yale. And that is where Dr. Rullo pioneered the development of synthetic antibody recruiting molecules that target UPAR expressing cancers. Dr. Rullo also serves as a scientific founder of Anna Biosciences, which develops next generation classes of synthetic tumor and viral immunotherapeutics. And this really uniquely leverages all of the tools he's developed in chemical biology with synthetic immunity. And of course, Dr. Rullo is also the director of training for the Center of Discovery and Cancer Research. And so I really want to encourage all of the trainees to come and speak to him. I think he's a fantastic role model for you guys and someone who's really ideal to serve in the position of training. He's not that far from his training days themselves, yet he's come so far. So let's welcome Professor Rullo. Yes. I don't know how to get that guy to go up.
Is it showing up on the?
Yeah, it is.
Paul, how do I get this guy to go up? I always mess it up. Last time it just went away. Where it says more?
Yeah, just more. There you go.
Just gotta click on this that is active. Good to go.
Well, my orchestrated joke slide just showed up 4 times now, so I guess it just lost a little impact, but that's okay. So thanks everybody for coming today. I'll be speaking about giving a general overview of the research program we have at McMaster that deals broadly at synthetically modulating cancer immune recognition using a strategy known as proximity induction. Okay, this is the idea of bringing things close together to enact a response. Okay, now this is attracting a lot of attention in pharma right now, especially in the United States, in the context of PROTACs, which some of you may have heard. Basically, bifunctional molecules that bridge natural machinery to affect targeted recognition and elimination. Okay, and so we're doing something like that in the context of immune oncology, but this is not real. This is actually quite disease agnostic work. Okay, so proximity induction basically is using these tools, in this case, a bifunctional that can bind to 2 different proteins at the same time. In this case, a tumor antigen, let's say that's highly expressed on a cancer cell, and an antibody that's naturally in your blood for example, can bring them together. This is a reversible binding process. That's why you see these arrows going back and forth. And in doing this, you can affect recognition by immune cells. In this case, let's say an FC receptor expressing cell like a macrophage or a natural killer cell, okay? But this could really be any cell surface receptor in biology, an endogenous protein. So you're basically using molecules that can control assembly of biological species, okay, to get an effect using chemical tools. This was an orchestrated joke. I noticed the last name was very similar. This is the Rullo French spelling, and this is the Irish spelling here. And so I just wanted to make sure everybody was in the right room because he is coming tomorrow. If that's where you thought you were doing today, I thought this is the first time I actually planned a joke in my whole life. So a little bit of context for where things started. A few years ago, I got involved in making these bifunctional proximity inducing molecules, okay, again shown here, where you have chemical ligands tethered through a linker that can bind natural proteins, all right? So in this case, let's call this an antibody binding terminus, and this a target cell binding terminus and a linker. What these molecules could do is they could bring natural serum antibodies to cancer cells, okay, that highly express a tumor antigen, shown here in red, all right? The idea is that you form this functional ternary complex, 3-component complex, and you form many of them at each location where there's a tumor antigen. So this works really well for tumor cells that express a high density of tumor antigen, all right? And so by recruiting antibodies to the cancer cell, you can induce targeted immune clearance and elimination of the cancer, all right? So synthetic bifunctional molecules that induce proximity, if you want to think of it in a fundamental way, of two proteins that are naturally present in the body, okay? One thing that we noticed looking at these in vitro and in vivo is that efficacy was limited. I'm not showing tumor model data, but you basically got, modest improvements in tumor regression, all right? And you got tumor relapse. So it could have been more efficacious. Even in vitro, we weren't getting 100% efficacy, we found, across multiple systems. This is an example. You titrate in different concentrations of your drug, in this case, an antibody recruiting molecule, and you only get fractional immune killing, in this case, cytotoxicity, followed by a decrease, okay? This bell-shaped dose response curve is classic in ternary complex pharmacology, happens for a variety of reasons that we can't really get into today, but just note that, okay, that we have a plateau that's limited and we have downward curvature at excess concentrations. This is known as autoinhibition or the prozone effect. This has been seen in heparin anticoagulation dose response curves, bispecific T-cell engager dose response curves, anything where you have to bridge things together, okay? This does not happen for typical drugs that simply saturate a target and form binary complexes. So we started thinking about, so when we came to McMaster, we started thinking about this on the molecular level. We thought about the bifunctional molecule, in this case, antibody recruiting molecule, as forming a reversible binding interaction with natural antibodies, okay? This is reversible and it's described by a dissociation constant, okay? A KD, which I think a lot of you are familiar with. And of course, if your molecule binds this with high affinity, then this equilibrium is going to move to the right and you're going to favor binding of your proximity-inducing molecule to the antibody, okay? At the same time, we imagined that this molecule is being rapidly cleared from systemic circulation, like a lot of low molecular weight species that partition in the extracellular compartment, okay, the serum. These things do not go intracellularly. So they're rapidly cleared. And so for ARMS to really maximize antibody recruitment to the cancer cell and form ternary complexes, you really are fighting a lot of natural barriers. You need really high affinity to drive these equilibrium to saturate the cancer antigens with antibody, and you've got your molecule getting depleted. So we thought maybe these molecular origins are responsible, at least in part, for the limited function of ARMS, okay, and proximity-inducing molecules in immuno-oncology. So taken together, equilibrium constraints, so these binding affinities, natural concentrations. You can't really control the concentration of cancer antigen or antibody in a patient's blood, okay? And so we thought about these and saw that there was intrinsic limitations in the way these proximity-inducing molecules worked, all right? And thought this is probably, any limitations here are likely extendable to other proximity inducing molecules that just don't have that name, like bispecific T cell engagers, NK cell engagers, et cetera. Okay. So we wanted to try and gain an understanding and develop tools to address some of these unknowns. Here are the key research questions that we started with. So one, are these endogenous species concentrations and affinities, so for the antibody in your molecule, for example, are they sufficient to maximize those ternary complexes on the cell surface, okay, that are required to engage immune cells and activate the immune system? The second major question was, when you have maximal complex formation on the cell, maximal ternary complex, is this sufficient for a prolonged anti-tumor response? Does that solve the problem right there? Okay. And the third is basically, how does the number of these complexes and their lifetime actually impact immune recognition? Okay, so some fundamental questions about how proximity inducing molecules work and how to enhance their function. So we came up with a strategy. Oh, looks like this is a bit too big. That we're calling covalent immune recruitment. Okay, that can be applied not just to antibodies, but recruiting any target protein in the body. So if you recall the scheme I showed before for ARMS, antibody recruiting molecules, what we did was we thought we could actually install unique reactive moieties in these bifunctional proximity-inducing molecules that allow them to not just bind, but now stick to their target protein irreversibly. So you can see now the arrow doesn't go back. In this particular case, this covalent arm can now link to the antibody permanently. We hypothesized this would allow us to gain control over how much ternary complex we formed on the cancer cell, okay? By overcoming these equilibrium constraints. So if you have low concentration of antibody in your blood, you can maximize binding through this covalent reaction, okay? This is just an example of the kind of chemistries that we use to affect covalent recruitment. So what are our goals? Basically, to use these reactions to irreversibly assemble different components that form these activated immune complexes to study the role of these interactions and try to ultimately generate design principles to get a stronger, better acting therapeutic, okay? On the fundamental academic level, we really see these as chemical tools to increase the stability and lifetime, okay, of these immune receptor complexes with tumor cells, all right? Because when you have the covalency, that interaction can't dissociate on that lifetime anymore, all right? And basically see how this irreversibility impacts the immunological function, okay? So this is a schematic of basically how a typical covalent arm works in the context of antibodies. You've got your bifunctional proximity-inducing molecule that binds, in this case, an antibody and a tumor antigen, but it has this reactive chemistry positioned in a certain way, okay? When it binds the antibody reversibly, at a certain rate, it will transfer, in this case, the tumor binding antigen irreversibly to the antibody. You can think of this as you generated a targeting antibody directly in vivo without engineering. It's another way of thinking about it, okay? So you have a covalent complex, irreversible. And you can actually, in theory, covalently modify any endogenous protein you have a binding ligand for. You can direct the chemistry that way. Okay, so the first thing that we did was we generated these covalent antibody recruiting molecules, okay, and we compared their binding and function to analogous non-covalent versions, so the traditional antibody recruiting molecule, the ARM. We developed some flow cytometry assays, binding assays, ternary complex assays that measure antibody recruitment to the cancer cell surface, all right? As you increase the concentration of your bifunctional, whether it's the covalent version or the non-covalent version, you get an increase in antibody recruitment to the cancer cell, okay? We used a specific type of antibody, in this case, anti-DMP, which is naturally present in the blood, okay? It recognizes this dinitrophenyl hapten. We increase the concentration of antibody and drug, and we get an increase in the amount of antibody recruitment to the cancer cell, all right? It's measured by a secondary fluorescent antibody. What we noticed was that the covalent recruiter increased the amount of antibodies recruited at a given concentration, okay? So this was in support of one of our hypotheses, okay? We're overcoming that first binding equilibrium. So at low concentrations of antibody, which are likely present in a patient's blood, you can increase the amount of it that's brought to the tumor cell, okay? And as we expected, at high concentrations of antibody, the covalency doesn't really matter because now you've got so much antibody that binding is now favored, saturative binding, okay? So this was actually very interesting and validating. What we expected to see now when we looked at functional assays was that at these low antibody concentrations, we'd see covalent recruitment give a functional enhancement, and at high antibody concentrations, they would be the same, okay? So if a patient has low concentration of antibodies in their blood, the covalency would be useful. If the target antibody was very high, covalency wouldn't be necessary, all right? Surprisingly, we did not see that. In black is Fc gamma CD16 alpha activation, okay, in a typical JERCAT bioassay used to validate tumor immunotherapeutic antibodies. When we do the same titration on the same cancer cells, by the way, these highly express the PSMA tumor antigen, but the tumor antigen isn't really, is kind of inconsequential for the point. As we increase the concentration of antibody and bifunctional, the covalent gives this pretty significant increase in Fc receptor activation, okay? We were shocked to see the reversible didn't give any. This is at across all concentrations that we screened, concentrations where we know even the reversible bifunctional saturated the cancer cell. So why is it that we've got a scenario where the cancer cell is saturated in antibodies, but the immune receptor is not getting activated? But when the antibodies that are brought there are linked covalently and can't come off, all of a sudden the immune receptors are getting stimulated. They're cross-linking, ITAM signaling activation. You know, as chemists and chemical biologists, this required us to really go into the immunology books and start learning about immune synapse formation and all the things that you guys know like secondhand knowledge, right? So this is the general idea. The antibody is now covalently linked. And what I'm talking about is that the reversible case had the same number of antibodies recruited, but they weren't held there. They can come on and off, on and off. The covalency actually allowed this to work directly using pooled human IgG, something that you would actually want this to be able to do in a therapeutic scenario, okay? So here we just took pan IgG and saw that our covalent bifunctionals were able to activate the CD16 receptor signaling, okay, in a dose-dependent way, whereas the reversible was not, okay? These are using natural anti-DMP antibodies that are already in your blood at low concentrations, as we found out, all right? The reversible just can't seem to do it. So that gives you an idea of the covalent enhancement. However, the problem's not solved. This y-axis, this could still be considerably higher from what we see with therapeutic antibodies, okay? So we thought this was really interesting. Another interesting thing we thought about this that we're actually pursuing is if we have all of this IgG present, okay, and only a small subset of it is the actually specific antibody we're recruiting, then these immune receptors should be saturated with antibody already. Why is it that we're able to actually get them targeted to the cancer cell? How is that possible using such a small amount of target antibody? There must be something more favorable when we're recruiting the antibodies to the target cell, okay, as opposed to all these antibodies just bind loose in solution to the immune cell. So this is a separate question that we're pursuing, okay? And I just wanted to kind of touch on it here in case anybody's seen anything like this in their studies, or especially in allergy and immunity, that these antibodies will saturate Fc receptors, but they're not binding a target. Once you have them, if you have them localized around a target, however, that multivalent display is very activating to the immune cell. So at this point, why does covalent recruitment make such a profound difference, even in vitro? Remember, this was largely designed to show an enhancement in vivo. We weren't really sure it was going to in vitro. You don't have clearance, right? You can use a high concentration of antibody, right? So what we saw was that covalent enhancement was maintained across all the antibody concentrations we tested. That antibody concentration range more than spans what you could find in a patient population, all right? We know we're seeing this enhancement even though the cancer cell surface is saturated with antibodies. This led to key questions. why is immune activation then sensitive to covalency? Okay, despite us having saturative binding. Saturative binding just means your concentration of target protein is much higher than its KD for the target. Okay. Basically, a very potent interaction. A second question came about if covalently linking to the antibody made such a difference, what about the interaction between the antibody and the FC receptor? Those aren't typically very high affinity, except in a few cases. Could that also be limiting the ability of these proximity-inducing molecules to enhance function as well? Okay, and would it be worthwhile trying to enhance that interaction? Okay, so I'm kind of priming you for the different directions that the program has gone in based on these original results. Okay, this finding led to these two models of FC receptor activation that we're currently studying in more detail in the context of covalent proximity-inducing molecules. So in this first one, we're thinking about it in a temporal way where the bifunctional recruits the antibody. These FC receptors have to cluster, okay? And this clustering has to occur over a certain time span or quick enough to activate signaling and have immune function. However, it's reversible. So maybe in the reversible bifunctional case, these off and on events keep resetting this activation response. It's just a qualitative way of thinking about it, okay? And maybe that's why covalency gave an enhancement, even though it wasn't recruiting more antibodies. It wasn't generating more ternary complexes, all right? It was basically preventing this pathway and allowing this clustering event to be enforced. The second one, which is a little bit easier to grasp, is this idea of strain and synapse formation. Around this time, conversations with the Bramson lab led to thinking about it in this context as well, where you have this mechanical strain during immune synapse formation and immune activation. We thought this strain would actually pull a non-covalent complex apart. So if this non-covalent bridging complex gets pulled apart, that synapse may not be able to form or mature to the point that's needed for activation. Maybe this is what covalency is doing, okay? By us locking in one of these interactions, we're giving it just that much more stability in the face of this pull. The reason why this is very significant is it tells you that even a strong high affinity binding molecule could still become quite weak in the context of strain. When we measure affinities, that's not under the scenario of any kind of strain, right? That's what these red arrows represent. If that's true, locking this interaction between the FC receptor and the antibody should then give you even more of an activation. Okay, so this is kind of the way we've been thinking about it and the way that we've moved. So these are the major questions at this point, basically for this FC receptor activation. Is it really sensitive to the kinetic stability of the complex, not just how many complexes, how many ternary complexes are formed? And the second is how limiting is this antibody IgG interaction? Okay. So kind of moving to an orthogonal system, some work that Nick Cerniak in the group and the Bramson Lab collaboration has been doing is looking at these covalent bifunctionals, not in the context of antibody recruitment to cancer cells, but in the context of engineered T cell activation. Okay, and so in this case, the molecule is the same, but now we have a whole different set of signaling interactions, okay, and things that are responsible for synapse formation. Engineered T cell, tumor cell, all right? We wanted to see if covalency showed an enhancement in a completely different system than FC dependent activation of, let's say, macrophages or natural killer cells. Well, lo and behold, when we look at activation of these engineered DAP12 SAR T cells, we see that covalency, if we're studying basically the number of cancer cells that are growing per unit time, these are LNCAP cancer cells that express the PSMA tumor antigen, okay? And so remember that these proximity-inducing covalent molecules can target antibodies to PSMA on cancer cells. In this case, the T cell was engineered to recognize our molecule, okay? This engineered receptor can now recognize the bifunctional, all right? So the bifunctional can covalently link to the engineered receptor directly. Okay, there's no antibodies here. Covalency gave you this remarkable increase in T cell killing of these LINCAP cancer cells, whereas non-covalency was basically overlapping with the controls. But again, we're forming the same amount of bridging contacts. Why is the immune cell getting so activated with the covalent version and not the non? We never anticipated this to happen. Another interesting thing, when we look at the exact same set of conditions with a different engineered T cell that has a different synthetic antigen receptor, this is the CAR, which is known to signal much more easily than the SAR, or sorry, than the DAP12 SAR. The covalency makes less of a difference. So there's something, this is in some ways kind of consistent with that second model I showed, this strain-induced model. If the synapse forms easier, it makes sense that it probably doesn't have as much strain that it's putting on that bifunctional bridging to enforce activation, okay? But if it's got a high threshold to be activated, the covalency might make more of a difference. Okay, so now kind of getting to the later second part of the talk. Now we also decided to initiate a project looking at direct engagement of FC receptors. Remember, our initial hypothesis was that if covalency made such a difference with antibody recruitment, it might make even more of a difference if we can avoid antibodies altogether and directly engage the FC receptor with covalency to study what the effects are. That's what this little schematic is showing. The covalent bifunctional, okay, in this particular case was with the antibody. Now we're doing a covalent linkage directly to this FC receptor, no antibody anymore, okay? What this would allow us to do is go after a whole bunch of different immune activating receptors on a whole bunch of different immune cells, okay? CD64, dectin, DC sign, okay? A lot of these recognize carbohydrates though, and not small molecule haptens, like the DMP we were using when we recruited anti-DMP antibodies. And a lot of these receptors recognize proteins, not small molecule haptens, okay? So we wanted to diversify this covalent immune recruitment technology to target a broader class of immune receptors directly so we could avoid antibodies and study what direct engagement of the receptor does to activation, okay? So to develop this strategy, we took antibodies as a proof of concept that recognize proteins or recognize carbohydrates, okay? So we could develop the bifunctionals against those. That's what we're talking about in these next two slides, okay? So Ben from our group has launched a pretty successful project going after rhamnospecific antibodies, okay? The idea here being that you could make the covalent bifunctional with a sugar moiety, so you can now target a sugar binding protein, like a lectin, okay? In this case, an anti-rhamnose antibody. And what we showed was that we can covalently recruit those antibodies. And since we're talking about antibody recruitment, we can measure that in FC activation assays or phagocytosis assays if we're using monocytes or macrophages. So if you look in blue, that's when we covalently recruit the antibody, okay, to the cancer cell. Really high phagocytosis was a function of dose. If you look at the reversible antibody recruiting molecule in green, it's way lower. And you're seeing this huge difference, again, for reasons that we talked about previously, and also because when you're using sugars, the intrinsic affinities are way lower. Sugars don't bind target receptors with very high affinity at all. So this bifunctional without covalency is even more limited than the series we showed in the previous slides, because we're using sugars, okay? And again, the way these phagocytosis assays are just double positive cell event reporter assays, okay? Cancer cells and immune phagocytes are stained with different flow dyes, and we look at double positives. These are just different prostate cancer cell lines that express different densities of the target PSMA tumor antigen. And these are HEC cells that are not engineered to express PSMA. So this is kind of your isogenic control of the top panel. All right. So now we have a technology to go after sugar binding receptors. Over here, protein binding receptors. Harrison in the group developed a really clever strategy where you could actually take a peptide epitope out of a target protein that's recognized by a receptor, or in this case, a virus-specific protein, an antibody specific for a viral protein. You can take a clip out of that, engineer it with a particular optimized chemical group to give you the covalent linkage. And now you can make a proximity-inducing molecule that recruits a protein-specific receptor or antibody, okay? So this is the general idea. This is a natural antibody that happens to be in the blood that recognizes this GD protein on HSV virus. He took a little peptide out that the antibody recognizes with modest affinity, and he engineered it with chemistry and a PSMA tumor targeting ligand to give you a covalent bifunctional, shown here. This binds the tumor, this reacts with your target protein receptor or antibody, okay, and this directs the covalency, this binding ligand. And what this enabled us to do was, again, stabilize these ternary complexes with covalency, okay? Only now we're not using a hapten. We're not engaging hapten-specific antibodies. We're engaging protein-specific antibodies, okay? And this just shows the selectivity of these bifunctional covalent molecules. LP4, these are fluorescence SDS page gels. If we look at the bifunctional molecule, we see LP14 is the monoclonal antibody that recognizes the GD domain of HSV, okay? And so the bifunctional contains a small piece of that protein, but the bifunctional can actually covalently link to that antibody now. And it contains a fluorophore, so you can actually watch that covalent reaction. So this is a band for the bifunctional molecule labeled antibody here, okay? And we see that when we use other IgG antibodies, you don't get fluorescence, you don't get covalent labeling. So that's how selective it is for its target protein, okay? And you can use competitors that knock out specific labeling and not specific. So a real successful advancement was made here because this now allows us to go after protein binding receptors, which sets the stage for direct engagement of macrophages, natural killer cells, maybe even T cells someday, all right? These plots on the right are just looking at the kinetics of how fast the bifunctional labels the target protein, okay, using different chemistries. The take-home message is this can selectively engage. The optimized versions can selectively engage the target protein within an hour or two, really pretty fast. Okay, that's fast enough to actually work in vivo before it's cleared and doesn't label other proteins. All right. So kind of the second last part that I wanted to talk about to give an overview was this idea that we've kind of been building towards, this idea that we could program immune cells directly, endogenous immune cells, not engineered ones, with targeting function, okay, and explore the role of covalency in immune activation. So just a brief overview here of our covalent immune programming strategy is basically CD64 on a macrophage can be selectively covalently modified with a targeting domain of your interest against the tumor, okay? Using this very specific bioorthogonal chemical reaction that I think a lot of you probably heard got the Nobel Prize in Chemistry this year. Because of the work that we've done up to this point, we can now covalently engage these protein binding receptors like CD64 that recognizes IgG antibodies, okay? And so now these approaches are being developed to target macrophages against cancers like multiple myeloma and GBM, okay, in the Sing lab. And what's nice about this is that you can actually take a biologic that's already approved, that already binds the target tumor, and simply program your immune cell of interest with it, either ex vivo or potentially directly in vivo, okay? This is showing a potential way of doing this directly in vivo, where the bifunctional molecule links covalently to CD64 on a macrophage and binds reversibly to an antigen on the tumor. Okay, and preliminary data is already showing us that we're getting more phagocytosis when we have this approach than the original, that was where the covalency was to the antibody. Now covalency is to the immune receptor directly, okay? From this, another project in the group that's gaining a lot of traction is this idea of not just covalently linking to one side, but both, okay? And so Eden in the group and Will have done some work on this, which is really cool. This is again is in the context of antibodies, but we want to use this to staple immune receptors to targets as well. Now you've got covalency to both ends, okay? So you can selectively bring two things together and keep them there. And we wanted to, again, study what the effects were on immune activation, okay? You can imagine this being applied to a TAC receptor or an engineered T cell receptor. Again, applying it to Fc receptors. Shown here is the CD16 activation potential. And if we just look in green, we're already seeing a functional enhancement when we can link both the antibody and the target tumor antigen together versus the original, which is in red, when we only covalently link to the antibody. And I should mention, this is not targeting PSMA anymore on prostate. This is targeting UPAR, which is a GBM target and a breast cancer target as well, and ovarian. In blue is when covalency is only to the tumor antigen, which I'm sure some of you have thought about by this point. Why just the antibody and not the tumor antigen? There's not a very simple answer for why you would pick one or the other. I can explain more another time. But basically, covalently linking only to UPAR, only to the tumor, and reversibly binding the antibody gives you a lot more of a functional enhancement than when you covalently link to the antibody and not the tumor. And when you do it to both, you get an additional boost. And I want to note that this boost we're seeing, the reaction is not anywhere near complete. It's probably only 10% conversion. So only a small portion of your antibody and tumor protein are completely covalently linked together here. If that was 100%, we would hypothesize that that enhancement would increase even more. Okay, finally, a platform in the lab that's been going on for a while, just a couple slides before we wrap up. This doesn't use covalency at all, but the theme of proximity induction is still there, where we're bridging immune cells to disease cells, in this case, cancer cells. So we talked about antibody recruiting molecules at the very beginning that recruited antibodies reversibly to tumor antigens, okay? One thing I didn't mention at that point was that those don't work very well when the tumor antigen expression density is low. And even monoclonal antibody therapies have this potential limitation, like Herceptin. If the HER2 expression is below a certain threshold, there's no more ADCP, ADCC. And so for antibody recruiting molecules, not surprisingly, we see the same thing. What was surprising was that we identified through cell surface binding studies that one major reason ARMs were not efficacious against these lower antigen expressing tumor cells is because the antigens are too far apart for the ARM to bridge the antibody through both Fabs. When the antigens are close together, for example, on a tumor cell that expresses a high density of antigen, these arms are close enough together where the antibody binds both through the two Fabs simultaneously. This is a very high avidity binding interaction, okay, which is much higher than the affinity for the antibody in this case. So the valency of the tumor antigen is actually perturbing the binding affinity for the antibody, okay? So when you have an antigen, a high antigen expressing tumor, you're binding the antibodies with a lot of affinity. It's almost like a covalent bond, right? When the antigen is low, the affinity for the antibody is much weaker because it's monovalent. So we thought, after discovering this, and this is work that Ben and Eden really led, we thought, well, maybe we can actually make multivalent constructs that have multiple copies of the targeting ligand for the tumor and multiple copies of the ligand for the immune component, whether it's an antibody or an Fc receptor. And we thought this would do two things. This would allow you to reach tumor antigens that are farther apart at the same time, so you could benefit from that high avidity binding. And also you could bring more immune components to the tumor, even when the expression of the tumor antigen is low, right? If you're bringing antibodies, you can only bring as many antibodies as you have tumor antigens, if it's a single molecule, right? But if it's a multivalent molecule, you can start to increase that exponentially, potentially, all right? So what Ben did was he made these polymeric molecules. This is just a little overview of the chemistry that's used to make these polymers, these multivalent immune recruiters. You can decorate these branches with tumor antigen binding ligands, and in this case, antibody binding ligands. This is that dinitrophenolhaptin that binds to anti-DMP serum antibodies, one of our model systems. Okay. And we can study binding to cancer cells and antibody recruitment and function using these multivalent systems. Okay. Now, our hypothesis was that we would be able to bind lower antigen expressing cancers, but with high avidity, okay, with a high overall affinity, and potentially bring more antibodies to these surfaces than you otherwise would be able to with a monovalent arm. So just a couple sample pieces of data. This here is a cell surface persistence assay. So we're looking at the recruitment of antibodies. We're looking at the recruitment of antibodies to these surfaces, okay, by these different compounds, all right? And what we see is a function of time, the molecule dissociating from the cell surface, right? It's a reversible binding interaction. What we see for the polymers in green, orange, and red, although red turned out to be largely nonspecific, we see that, you know, a very slow half-life. By 24 hours, we still have 50% bound to those cells, all right? For the monovalent antibody recruiting molecule, it falls off much faster, okay? And this is consistent with your antibody recruiting molecule being able to bind the target with very high avidity, okay? Or sorry, the monovalent can't, the polyvalent polymer can with high avidity because it can reach those farther antigens at the same time. This is now looking at differences in antibody recruitment, okay? So if we look at these lower antigen expressing cancer cells, these are PSMA expressing LINCAP and C4-2. The polymers can recruit a lot more antibody to the cancer cell compared to the monovalent arm, okay? So remember, we wanted to amplify antibody recruitment to lower antigen expressing cancers. The polymers look like they're actually able to do that. So it's a really nice proof of concept here. And this manuscript is now in review, which is really cool. So I'm just going to cap off the presentation with some take-home points, what we've learned. So what we've learned so far is that covalent recruitment of antibodies to tumor cells does enhance FC activation, okay, leading to anti-tumor responses. We saw that we could adapt these covalent immune recruitment strategies to different immune receptor targets, different antibodies. We also see that it looks like immune synapse formation may show the sensitivity to the kinetic stability of these bridging complexes, okay? The fourth big question here is, now, does immune synapse formation actually impose strain on these bridging complexes, okay? That promotes dissociation of the therapy. So this is something that we're working towards now using microscopy and maybe potentially some molecular tweezer experiments. So we start to go more into the realm of physics to try and address some of these questions. And lastly, when we directly engage these FC receptors with these covalent proximity inducing molecules, will we actually get an additional enhancement in anti-tumor function as we hypothesize? And so that's it. I want to thank the students in the group for a pretty heroic effort over the past few years. We landed in 2018. Nick, who was our first with Eden and another student, and Ben kicked off this covalent antibody recruitment work, okay? Harrison and Cece then joined and have already been making great contributions to the group. Thank you to these funding agencies for taking a chance on new investigators without much preliminary data, which isn't something a lot of people are willing to do. Our collaborators across Merck and the CDCR, and these are my three daughters that I just put in for awe factor, because objectively, I think they're stunning. Thank you. That's it.
We still have time for questions. Maybe first trainee, if you have any questions.
For your have you guys thought about any having on-target off-tumor, especially how we build it up?
Absolutely. It's a trade-off. Like anything that you do with an immunotherapy, for example, right? You're always navigating this balance between inducing a self-reaction versus a targeted anti-tumor reaction. So there's, what's that? Say it again, sorry? You could get at it by really carefully controlling dose and a finely tuned therapeutic window. We actually have some chemical strategies that we're working on in the next gen of those polymers where we can pre-target the tumor, but there's no immune recruiting ligands, and then we can use bioorthogonal chemistry to take monomeric immune recruiting ligands that won't activate anything in systemic circulation because there's not a clustered presentation of them. And then they click to the scaffold and you only get that amplification at the site. Little strategies like this we're working towards. Thanks for the question. What's that?
I don't know anything about chemistry, so I'm really naive. But in the context of T cells and maybe other immune cells, the distance of the synapse is possibility of something that is particularly important. Have you guys ever noticed or looked into your different chemistry, whether they increase or decrease the depth of the synapse, and whether that would affect the strain?
No, it's a great question, and that's a very important variable that we are in the process of screening as well. Basically, how close we're bringing the targets, how compact these bridging complexes are, because it's been shown, especially for bispecific engagers, I think that distance between the target and effector really affects the synapse formation and impact. Is that along the lines of your question?
Yeah, because I was kind of thinking, again, I don't know anything about.
Yeah, they're pretty small
Yeah.
compared to a protein.
There's a long one. Remember the next one's really long. You get a long kind of.
Yeah,
It looks big on a screen, big in real.
yeah. In real life, it's pretty small compared to the protein in most cases.
Okay.
But, playing around with the rigidity of these and the length and looking at impact on function is definitely on the to-do list.
It also truncate the, like, truncate the star back. If you do have, multiple types.
Yeah. No, it'd be a cool thing to try.
Maybe I talked about it.
Yeah.
Yeah, any other question from, yeah, Ali?
Yes. Yeah. No, absolutely. And this is actually something that Professor Ackar brought to my attention a couple of years ago. In the context of CD16, yes, that would be problematic therapeutically. And I know that a couple of the folks that are engineering bikes in Minnesota, you probably know them. Can't remember the name. They just started a company, I think. They're very concerned about that. And one thing we were thinking about, it's just on the intellectual side, was if we could actually connect covalently to that cleavage site. So the protease can't cut it, but you can only just reach so much in so much time. But yeah, it's on our radar for sure. We're more interested right now functionally in macrophages, and because we can't compete with Ali. So
You can synergize.
we can synergize. Yeah, so that's a great point. What was your second question? Yeah, no, absolutely. I think there's, a lot of these are kind of meant as tools to basically learn and see and kind of understand how the system works. So findings like that would be very, very cool as well. I don't expect the majority of these to end up in a therapeutic context. I mean, the dual covalent, it would be very interesting to see what something like that does, because there's a lot of implication. I mean, it's not a natural thing you've done, right? So you're going to expect to see, that's one potential thing that could happen. We've been imagining pretty big implications on turnover events, for example. I think it's just gonna be really cool to see what happens. And, you know, at the end of the day, we can always take it and try to modulate gene transcription, for example. You know, so it's kind of target agnostic at this point as well. But those are great points.
Any other question from anybody? I think you're a participant in online as well. I don't know how you can, maybe we just probably stop sharing so they might have questions
Yeah, absolutely.
in case. I think they're all. Stop sharing.
I also wanted to thank the trainees. I noticed that all these posters, not one was vandalized. That was really,
Yeah, if any of the online participants, if you have any question, please. Go ahead. If you have any.
let's see in the chat. There's one in the chat box. I see perfect, but I don't think he means the presentation. I don't think Jim means the presentation.
If not, I have a question, if nobody has a question, because I think you asked this question from your student, and I think it was unfair because you were not there. So maybe we just grill you here.
You want to get
on
me
that
now?
Question.
I have it coming.
So I think the question about macrophage is very interesting because macrophage in solid tumor microenvironment, they are becoming very much friend of the tumor.
Yeah.
And the concern that we asked one of your students at that time, that maybe this engagement really would stimulate them to produce the factors that already tumor-like. And the question would be that, do you think this engagement would really turn your macrophage away from pro-tumor to anti-tumor, or like Treg, for example, that right now the Treg engagement, you can engage them with any engager, but it's still they do Treg function. So the question would be, maybe if you engage those TAM or tumor-associated macrophages, they would do the job that they are supposed to do.
Yeah, I think that's a great consideration. And, you know, it's something we're gonna have to test. There is precedence that it depends on what receptor you do engage. For example, the dectins have been shown to, if a certain multivalent engagement of the dectins can change the polarization state of the macrophage. Therapeutic antibodies like monoclonals have shown to change the polarization of TAMs as well, which kind of had us think that maybe covalency would give us an additional enhancement, but it really has to be tested because, as you say, that's a possible effect.
And I think Dr.
Tobias, could combining this macrophage engaging strategy.
Tobias, do you want just ask a question, or maybe?
Tobias, do you want to talk out loud, or do you want me to just?
Yeah, can you hear me?
Yeah, I sure can.
Oh wow. Okay. Very nice talk. Very nice. Interesting strategy with engaging these macrophages. In the AML world, there are people looking at these "don't eat me" signals, because they found those important originally in trying to understand which mouse strains take AML cells and which don't. And then they design strategies against, for example, CD47, which is essentially like when this is on the surface of a tumor cell, this essentially tells the macrophage, "don't eat me."
Yep.
And so could combining your engaging strategy with an antibody against the "don't eat me" molecule be kind of a synergistic combination?
I think it would be, and I'm aware of CD47. We have a project right now with a private partner that involves CD47. But yeah, no, I think that would be a great thing to synergize, to actually get some of these things to have an actual a real immunotherapeutic application. I think that would be very advantageous to engage that. Yeah.
Great. Any last questions? Yeah.
Have you thought about tuning CD64 and engagers to actors on the protein?
To try and induce degradation? No, we haven't thought much about the degradation space. Only because and I should mention, recently, the degradation space has started to adopt this covalent recruiting angle. And so I imagine that people are going to be trying this probably pretty soon. There's already covalent protacs, for example. Yeah, I mean, I think in the context of neurodegeneration, it might be something that I'd be interested in pursuing. I'd have to see what the group thinks also. You know, it is a democracy. Yeah, it's definitely something that's interesting. And it's just kind of, you know, weighing those pros versus cons. You know, do we want to basically learn how to do those degradation assays, bandwidth, those kinds of things? But I mean, in theory, absolutely. Yeah. Yeah. No, absolutely. Well, the thing with LITAX was done by the Nobel Prize winner in chemistry this year. That's another thing to consider is if we were to pursue this, you know, it's there's kind of a strategy consideration as well, right? We probably would get mulled initially. Thank you.
Go ahead.
Yeah, well, absolutely. You know, a lot of what we're talking about, it's a reductionist approach, and then we build complexity from there, right? And so everything that we're showing is kind of you know, we're not assuming any of these other effects are at play when we make predictions. But of course, you know, something like pH, you know, if it alters the conformation of the target protein, could be very consequential, right? We've thought about using these tumor environment features to activate release of certain cargo, as has been done with antibody drug conjugates in a way. But yeah, pH, hypoxia, steric occlusion, right? We think about smaller molecules that can penetrate better. These are very important considerations for sure. There's all kinds of enzymes that are secreted that we can use potentially to our advantage, but the molecules themselves are fairly inert to any kind of biological protease or pH. So that part shouldn't be too problematic on the chemical end. But yeah, it's a very valid point. What we've talked about so far today is they're also not orally bioavailable drugs, right? These would be most likely IV type. In theory, they could be constructed to be orally bioavailable if that was advantageous, but these probably wouldn't be absorbed very well, what I've shown today at least. The sugar molecule that I showed, that would be quite pH sensitive actually. So that's a good point there. That could get cleaved. Yeah, so it's an important consideration in general.
Okay, if there's no more questions, please join me and thank Dr. Rullo for a great talk.
Thank you, sir. Thanks for holding the torch.
You got it.
Thanks, everyone.